Before the lightning talks get started, I just wanted to iterate that these lightning talks are for you. These lightning talks are not really directed to ARPA-H. They're for one another, everyone who's online and people that are here, because we want you all to meet each other and understand what capabilities different entities have. And there'll be many more opportunities for doing this beyond the lightning talks. And the last thing I wanted to reiterate, please read the solicitation. I saw a lot of Q&A come in where it was clear that not everybody had read it. A lot of people worked really hard on it to get it right. So make sure you do that. Thanks.
All right, my name is John Douglas with the Healthy Buildings team. Our team does three roles. We keep up with the science and technology related to infection control in buildings and indoor air quality and the benefits of indoor air quality on our building occupants. Microphone. Oh, microphone. All right. Take it off. All right. I'll just stay here. And then the second thing we do is we assess technologies. So we meet with a lot of third-party vendors to see what technologies would be good for buildings and assess them. And then the third thing we do is we put all that together into a solution that we offer to our customers. And what we found over the years is that the solutions that are directly related to a known customer need tend to be the most successful. And that's so much so that our greater JCI team actually has customer feedback sessions where they bring in customers 2 times a year. For example, we have a hospital session where we bring hospital administrators in, ask them what are their needs for buildings. They give us feedback, and we use that to develop solutions. So what I want to do real quick is give you a couple market feedbacks and show how we threaded a solution through those needs. So this is first one. When we think about healthy buildings, it's really the intersection of 3 important things. One is clean air, indoor air quality, sustainability, and then the occupancy of a building. And so if you think about it, we all know about clean air and the benefits of clean air. But if we don't do it in a sustainable way, we're going to create an environment that makes that indoor air even dirtier. So we got to think about that. And that's also a pressure the building operators have in their building. And finally, COVID has changed the way we use buildings. And so occupancy has changed quite a bit. So this next one is sensor data. Hold the mic. I keep walking. So sensor data. What we found when we put a lot of sensors in buildings, and for our customers, just simply reporting values is not enough. Those values have to drive an operational change. People don't want to know, hey, your PM is high. They want to know your PM's high, and therefore you should blank. And this is a chart that shows the different contaminants in buildings. There's gases, particles, and pathogens. And then there are different means of getting rid of them. Okay, outside air ventilation, filtration, and you can see then we've marked the sensors. And if you note in the bottom, and the reason why we have the ARPA-H is if you look at disinfection, there's no sensors for it, right? And that's a big problem because we can't measure and choose to see if we're doing disinfection. So we put this all together in a quick solution where we do IQ auditing. We take CO2 sensors, we can detect occupancy and compare it with the building schedule. And we can then quickly adjust the building schedule just using simple control set points to make sure that the building is running and using that energy when the people are there and providing indoor air quality benefits. And that's it.
Thank you. Next up, we have Eric Malmstrom from Safe Traces.
Good morning. Welcome to Oakland. Thank you for scheduling this here, given that I live here. So I'm going to be giving a talk that's called Reality Check, the Role of Technology in Breathe. So I'm going to start off with 3 big, hairy questions that I think we should all be asking ourselves. The first is, how do we know biosensors are working? The second is, how do we translate between what I call health and building? And then third, how do we know building controls are actually working? Where our company, Safe Traces, comes into this is we have a decade of experience coming out of Lawrence Livermore National Labs and then in the commercial world, basically using an aerosol pathogen surrogate to test buildings and what's happening in the buildings, how are the controls working. We've done a lot of research, we work with top experts in the world, and we've been out in many buildings, millions of square feet, everything from Class A office buildings to prisons. And we've seen a lot of stuff. So that's where kind of my perspective is coming from. I've got bad news and good news. Nothing says bad news like someone crying. The first is that IAQ sensor performance of the sensors themselves and the data and the data quality is highly variable. And we see that all the time. Biosensors are taking it to the next level. It's much more complex. And so we should be asking ourselves, are those sensors working and is the data quality strong? The second is biosensors speak the language of dose exposure, but building controls speak the language of HVAC, which is MERV 13, outdoor air. And those are not translatable languages right now. We're starting to get there, but they're two different things. The third is automated building controls are limited and not focused on health, but also we should be clear, building controls in many buildings are humans. 15% of buildings have BMSs and they're pretty unreliable. Good news is tech is closing gaps. We're seeing the beginnings of a translatable language between dose exposure and HVAC and plus other interventions like UVC. And finally, controls and the operators are being forced to contend with health considerations more and more. Where we come in is we basically use our challenge agent and the software platform behind it to reality check the biosensor, to calibrate them, commission them, act as a control, and then also to ensure that we don't have the garbage in, garbage out problem of the data. We then translate that data from dose exposure to building control language at portfolio scale. And third, we then provide that data to the BMS and to the frontline operators to make sure that they're intervening and making the right types of decisions. Thanks so much.
Thank you very much. Next up, we have Lindsay Marr from Virginia Tech.
Hi, I'm going to talk about lessons learned from Mitigate Flu, which is a program or center that I direct. It spans the 3 technical areas or parts of them. We have a project that focuses on detection of flu virus indoors. We have animal models where we're looking at transmission and mechanisms and interventions, and then also a study of humans where we're looking at health outcomes. The first lesson learned is that sample collection is rather challenging in occupied spaces. We might be interested in collecting, detecting a concentration of 10 plaque-forming units per cubic meter. If you breathe that for 15 minutes, you get a dose of 1 PFU. So you need to be able to collect that air, get it into liquid usually for detection. High volume sampling pumps are noisy and you can't use them in occupied spaces continuously or at all. So we did this as part of a Well Complete project looking to develop a rapid sensor for SARS-CoV-2 in the air. We didn't quite get there, but I want to say that WashU has a solution for looking at SARS-CoV-2. They have a few posters here. The second lesson is that despite that, we were using traditional methods over the past 2 years, and we do find respiratory viruses in air and surface samples in childcare centers in Michigan. We have 340 air samples over that period, and we detected flu A and B, SARS-CoV-2, RSV, and rhinovirus in those, about 3% of samples. The concentrations are around low hundreds of gene copies per cubic meter, a couple instances in the thousands of gene copies per cubic meter. Our third lesson from our project involving animal models is that when you have lots of close contact, ventilation has limited effectiveness on transmission. We did a study where we have one donor ferret that's shown with the green, so it's inoculated with influenza virus. We exposed 4 naive ferrets to them under conditions of low ventilation and high ventilation, where we have 1.3 and 23 air changes per hour, so a difference of more than a factor of 10. In the low ventilation case, we saw transmission to 4 out of 8 ferrets, or a transmission efficiency of 50%. In the high ventilation case, we saw the same outcome, transmission to 4 out of 8. The ferrets were allowed to play together and interact. This is simulating the type of behavior that we think we see in childcare centers. Finally, we are evaluating risk of transmission in a human challenge study where we have an aerosol inoculated donor and then 3 naive recipients together in a small chamber. This allows us to study dose response and also the effectiveness of interventions in a very controlled setting. So we believe that we have expertise in detection of virus in environmental settings and also understanding of transmission that can help Breathe succeed. Thank you.
Thank you very much. Next up, we have Albert Karam from PCCI.
Okay, so I'm actually Mike Pompous. I'm from Harvard University, and I'm here on behalf of the Harvard University California Irvine Infection Prevention Group. We are 3 professors of infectious diseases, population medicine, epidemiology, And this is what we do. First of all, we're clinicians. We take care of patients with infections. And so we understand what it is when a person gets infected, how that happens, and what to do about it. We run hospital infection control programs. I run the program for the Brigham. My colleagues run for UC Irvine. And we conduct high impact research on infection prevention and control. We've done a lot of work on cluster detection and control, transmission detection and prevention, improving diagnostic clarity. And we all help set national standards in infection control. My colleagues and I, we sit on the panels that write the guidelines. We talk to CDC, we talk to professional organizations. Much of what's considered standard practice comes from my colleagues and myself. Here's an example of some of the things we've done, of some of the papers we've put into top flight publications in the medical space. It's a combination of editorials suggesting what we ought to be doing, as well as large cluster randomized controlled trials in dozens and sometimes hundreds of hospitals or nursing homes, looking at decolonization strategies and their impact on actual infection acquisition. And these are the kinds of studies that in many cases have driven changes in policy, changes in guidelines as to what constitutes an order of practice in infection control. What we are offering is clinical and operational expertise. We have a sense of what's clinically meaningful to doctors, to clinicians, to patients, as opposed to simply looking at proxy markers, and what those sorts of things that are needed to make a hospital or a nursing home ready to accept a technology. What are the sorts of things that, what are the sort of design features that are needed to make it realistic that one of these institutions will institute this? And what are the sorts of impacts or outcomes that they want to see in order to benefit, to see a demonstration of benefit? We have expertise in trial design and conduct. We've been able to design the trials and to conduct the trials that have led to practice changes in standards around infection prevention, around decolonization, et cetera. And we have partnerships. We have partnerships with hospitals. We've partnered in the past with the Hospital Corporation of America. That's one of the over 140 different hospitals with large nursing home networks in Orange County, California, and elsewhere in order to implement these projects. And so if you have a promising technology that you feel is ready for the prime time, we can help you to refine it, to work out what it needs to actually make it operational and acceptable to a real life healthcare facility, and then design with you the trial to actually test it and show the world that it works, and then collate for you, distill for you your trial experience and present it in a top flight sort of a publication so it has the possibility to have that large scale impact. So if that's for you, reach out to us. Thank you.
Thank you. That was Michael Klumpus from Harvard UCI. Now, next up, we have Albert Karam from PCCI.
Afternoon, everyone. So who is PCCI? We're the Parkland Center for Clinical Innovation. We're a 501c3 nonprofit in Dallas. We focus on predictive analytics, AI, generative AI, and implementations of those analytics, specifically for hospital settings, payers, but primarily it's for the county, it's for the groups, it's for the people who live in those areas and facilitate all of the care that happens there. One of the things that we have is a pediatric asthma surveillance system that we created. We've extended this to diabetes and hypertension as well. But the key factor here is the non-medical drivers of health or social determinants of health. We focus on providing a whole health approach, looking at things that are not just in the physical hospital or in those settings, but in the community. One of which is outdoor air quality, which we focused on heavily to understand the risk. So in risk assessment, we want to look at the whole person, where they come from, where they play, where they work, where they live, where they pray, and we want to be able to identify those types of trends in the communities. Don't really need to preach on this. Why is this important for indoor quality, indoor air quality? People are inside. We've heard enough about that. One of the experiments that we did was an indoor smoking experiment. We don't develop the sensors, but we do risk assessments of them. We set this up for the underinsured, lower insured groups to try and help mitigate asthma and other chronic diseases, particularly in children. So we set up air quality sensors in the houses. We did some tests where we would smoke in one room, see how it affects their asthma, see how it affects the other rooms, how far it can go. You can see, really easy spikes. One of the things that we did was we hooked it up to Alexa, and we had Alexa be able to understand if the sensors were hitting certain thresholds based on the socioeconomic factors, the medical factors, and the air quality sensors combined. And then it would start talking to the person, trying to provide opportunities for them to get to a better place with better air. Ended up not getting funded, but it was a really cool test project. We think that we provide a robust approach to AI and ML. It's not just one thing. It's not just one algorithm. The outreach needs to span multiple different domains. With us being in a healthcare setting primarily, we need to make sure we address the provider. We need to address the schools. We need to address the payers. We need to address the patients. And we need to have outreach in each of those areas. The most effective way that we've found has been text message. Not everybody has a smartphone. If we're trying to do a program that impacts everybody, text message has been the best way for us to get that feedback loop from the individuals, and that's been our approach. Thank you so much. Feel free to see our poster.
Thank you very much. Next up, we have Sam Molnix from Poppy.
Thanks so much. So I'm incredibly excited about this program, as is the super strong team that's formed around us at Poppy over the last 3 weeks leading up to today. So a little bit about us very quickly. That didn't come out right. So we're a venture-backed technology company located in San Francisco and Toronto. And our mission is to build healthy and sustainable buildings using advanced air technologies that can enable suppression of infectious aerosol transmission over space and time in buildings. If that sounds familiar, that's really the vision of this program, Jessica's remarkable vision. And we've been pursuing it with intensity since 2019, where we've developed a significant technology and patent portfolio around this. We've been able to scale our services to 25 cities, 1,000 locations, on land, at sea, marine applications, schools, sort of every imaginable space type. And we're from some of the best tech companies and institutes out there. I previously co-founded Meta, which was an early artificial intelligence company, spent some time at Chan Zuckerberg Initiative, and then co-founded Poppy Health in 2019. And the work that we've done as a team has been mentioned in over 150 publications worldwide. So hopefully these slides can be made available because there's stuff that really didn't come out somehow. So in essence, Poppy has a stack of technologies that comprise infectious aerosol detection. We actually ran a live pathogen detection network through the pandemic for several years, 30,000 SARS-CoV-2, flu and AAB and RSV detections. And what we learned through that is that actually aerosol tracers as a backbone technology for both calibrating your HVAC system to enable equivalent clean airflow for detection of or protection of people in indoor environments, and calibrating your biosensors or biodetection system is critical. We invented a series of technologies around these, including massively multiplex DNA barcode, aerosolized tag detection, and real-time air tracing. And part of what you'll see in some of the technologies that I'll sort of glimpse to you quickly right now gives you a couple of sort of hints around that. We're an incredibly strong technology integrator. You can't see it on the slides here, but we have machine learning, data science, cloud computing, rapid hardware development, really all the pieces that are needed for this project. So here's a couple of views of our amazing technology. You can see infection vectors over space and time in buildings. These are where you put your biodetectors. Here's what the ASHRAE 241 framework looks like as applied in a real-time installed system in buildings. We have these running in whole towers in downtown and cities. Here's another example. You can see infection vectors, calibrate all of your systems against these data. And here's what the variability of these systems looks like over time, why it's incredibly critical to be able to run these in real time. So I'll just show you something very quickly at the end because my slides didn't work, but we have an incredible team coming together around this. We've put together the infection risk modeling group from 241 for TA2. We're partnering with Schneider Electric and Daikin, two of the global leaders in HVAC systems, to deliver TA2 and TA3. And we're looking to partner with great bioaerosol teams who have sensors that have some sort of clear scaling to 25 plex as part of this program. So get in touch with us and I'll put our information on the website. Thanks.
Thank you very much. Next up, we have Andrew Koblinski from Primary Health.
Welcome. All right, let's get into it. So Primary Health we're a group of software developers that started as a volunteer effort in a pandemic, and it became a company in response to public health needs. So we've always been focusing on what are the unanswered questions and needs of underserved populations. So our software platform has been used for a lot of different studies. Early in the pandemic, UCSF, we did the data work behind the first studies that uncovered asymptomatic transmission of COVID. And throughout the pandemic with CDPH and other organizations, really tried to answer questions. We also spent about $1 million of our own funds trying to understand what comes next after large mass scale antigen testing programs as our mitigation measures. What can we do with improving air quality? So we funded several projects. I'm going to talk about just one of many here in Oakland Head Start locations. But before I do that here, our capabilities are in TA2 and TA3. We're mostly focused on the software of understanding the data and software also about patients. We're high trust certified. We work with patient information every day. We've run millions and millions of tests, millions and millions of immunizations. We're used by many states as statewide immunization software platforms and testing platforms. We integrate with public health labs, et cetera. So we bring a lot of capabilities and deployment experience in the real world. So in Head Start, to give you an example of a full system, and I would love the ARPA-H brief because we actually did like a mini version of this to try to understand how to deploy something like this. So we conducted pathogen detection. We did air traces. We worked closely with Poppy and various tools over the years with them. We did iterative improvements, education, people, interventions, testing, et cetera. We did everything and we learned and repeated and changed and learned and repeated. So what that looked like, some of the tracer tech from Poppy, leave behind sensors to do passive Wi-Fi enabled, pathogen detection, lab-based detection, pulling samples to a lab for PCR, getting the results, turnaround time, trying to impact policies and changes. Rapid antigen testing in combination, some mitigation programs, masking protocols, et cetera. During the Omicron surge, what was most interesting is that we learned the best way to clean the air in indoor spaces was to stop the introduction of spreaders. So what we found was, even though you may improve air quality in spaces, using those mitigation measures to stop spreaders from entering those spaces is the most effective, as Lindsay expressed earlier. That, yeah, in childcare centers, that's necessary. So we have a lot of findings, implications. I suggest you read our slides. At the end of the day, the tech solutions are half the problem. Deploying in the real world is the hard part of the puzzle. So I love that this brief is challenging us to get real world solutions done and created. Thank you.
Thank you very much. Next up, we have Connie Chang from the Mayo Clinic.
Hello, everyone. I'm Connie Chang, and I'm an associate professor in biomedical engineering at Mayo Clinic. I'm here on behalf of my team here, James Wilking, Kui Tao, and Clifton Hayter. Just to give you a little bit of an overview of Mayo Clinic capabilities, we are mostly focused on TA1 and TA2. In TA1, we have quite a bit of expertise in droplet microfluidics for pathogen detection, also point of care diagnostics, highly integrated electronics. In TA2, as you know, Mayo Clinic, we are a large hospital. We have a lot of clinical expertise, experience with healthcare data, and IRBs. And then we have 3 different locations in 3 very different climates in Minnesota and Arizona, and also Florida. And we have a mobile health clinic that serves over 2,000 patients in rural communities. Within droplet microfluidics, we have a way, so droplet microfluidics is a way of doing high throughput screening and assaying of pathogens at the single virus, single bacterial level. We can actually multiplex and detect these pathogens and detect up to hundreds of different pathogens. And finally, we have experience with point of care diagnostics and being able to test and commercialize some of these microfluidic devices. I don't know why the slides are a little bit mixed up with PCCI, but I'll continue. And then also within TA2, Kui Tao is the chair of our bioinformatics and AI department. And the team here has a lot of experience with social determinants of health, also with the exposome and AI-based risk prediction. So things like looking at maybe how likely you are to get another asthma attack, or how likely you are in terms of location to have access to healthy food. And finally, we have expertise in the health Internet of Things, so micro-miniaturized, scalable, flexible, real-time ML AI. So this is integrating with a cell phone to be able to transmit wearable devices data or data from ambulances. Okay, and as I mentioned, we have 3 different sites, Arizona, Florida, Minnesota, the mobile health clinic, close to 10,000 active studies reviewed by our internal institutional review board, and one of the largest clinical laboratories in the world, performing more than 27 million tests and pathology services per year. And we are mostly looking for partners in TA3, but also open to partnering in the other TA areas. So thank you very much. Oh, okay, yes. So I'm Connie Chang, biomedical engineering at Mayo Clinic.
Next up, we have Jeffrey Schroeder from ARA.
Hello, thank you, everyone. Applied Research Associates, or ARA, we're a medium-sized employee-owned company with offices throughout the country. But our small group or project team within that company is in the computational modeling and respiratory dosimetry group. We have offices in North Carolina and the DC area. We have a multidisciplinary team consisting of aerosol scientists, software developers, and so on. We mainly work on projects involving human health impacts from inhalation exposure. And so we develop quantitative models and software platforms in support of those assessments. And so we're, of course, looking at considered for TA2 respiratory risk assessment and looking to partner with other organizations for TA1 and TA3. So the next couple of slides, like to give you a quick overall background of some of our relevant experience. We've developed multiple software platforms for respiratory risk assessment for various government health agencies, such as EPA and FDA, other agencies within DoD. We do a lot of work with exposure dose response modeling. One of the fundamental premises of much of our work is that exposure response concentrations are usually not sufficient for risk. Internal dose in the respiratory tract is a key contributor and main driver of risk. We've also looked at and many others have looked at machine learning models for various applications. I think ML is going to play an important role given the large amount of data to be generated in these programs. We've also done some work with indoor air simulations, CFD and building and such, and also person-to-person transmission of respiratory diseases, specifically following the COVID outbreak. Some specific examples of respiratory risk assessment software. We have the publicly available multiple path particle dosimetry software that we've developed. This is currently being used by EPA as a program of record in their inhalation risk assessment. We're also currently developing a risk assessment tool for the FDA Center for Tobacco Products using dose response and other relationships to quantify human health risks from tobacco product constituents. And we also have other software platforms in development at the EPA, such as Track My Air and Glimpse, that do various air quality and environmental planning. So we do have a poster. Be happy to talk with anyone about more details about those software platforms. And thank you very much.
Thank you very much. Next up, we have Meng Kong from Well Living Lab.
Okay. Hello, everybody. My name is Meng Kong. I'm the research lead of Well Living Lab. And our lab was founded as a collaboration between Delos Living and Mayo Clinic. And since founded, we have been working on research about indoor environment health. We are a highly collaborative and interdisciplinary team with scientists from both building science and health science background. And we indoor air quality and building environment and system control are the 2 biggest topics of our research. And our lab has the capability to support both the technical area number 2 and number 3. And more specifically, and for TA number 2, we have the expertise and experience in the exposure response experiment, in infection risk model, and the software development. And we have design and manage and complete several studies that looks into the impact of environment intervention on people's health, performance, and behavior. And also during the pandemic, we have conducted several studies on the distribution, deposition, and disinfection and cleaning of indoor aerosol, and develop a tool and software to predict the indoor COVID exposure risk and the performance of engineering approach in terms of mitigating the risk. And for TA number 3, we have the experience and expertise in the sensor and product development and testing and integrated building control. We have developed several environmental sensor and wearable protective equipment, and currently are involved in the development and testing of a platform and hub for which actually integrate multiple environment sensing technology and integrate into the building system control. Also, we have completed a study, ARPA-H studies before, within which we are responsible for testing and evaluating the environmental and energy impact of occupancy sensing technology integrated in the building HVAC control. And we have a poster in the post session. And if you are interested in our lab, please feel free to stop by. And thank you very much.
Thank you very much. Next up, we have Peter Wynn from the Weiss Institute Collins Lab.
So just give you guys a quick introduction to the Wies Institute. If you've never heard of the Wies Institute, it's Harvard's Bioengineering Medical Institute. And we do a lot of things, kind of outside of the box things. We do robotic bees, organs on a chip, super tough hydrogels, injectable vaccines, DNA origami, gene editing. So if you've ever heard of resurrecting the mammoths, that's us as well. We also do a lot of synthetic biology. And within the Wies Institute ecosystem, I'm in the lab with Jim Collins. And Jim Collins is one of the pioneers of synthetic biology, created one of the first synthetic gene circuits 24 years ago. And we do a lot of work in molecular diagnostics, specifically nucleic acid diagnostics. So our focus is going to be on TA1, and we're looking for a team with TA2 and TA3 expertise. And to give you guys a snapshot of what we can bring to the table, this is our kind of the greatest hits in terms of the molecular diagnostic space. And so our technology, what we can do, our expertise is to take biological reactions, sensors, and actually freeze dry them. So you can turn it into a powder. Now it's freeze dried powder. You can just add water to it anytime you want, and it's stable for up to a year. You can store it at ambient temperatures. It doesn't need to be refrigerated. And then now this reaction can actually detect any nucleic acid in the environment. And I'm just going to run through a couple of our papers. One of our papers back in 2014, we developed these freeze-dried sensors. We put them on paper. You can see it right there being held. It's about the size of a stamp. It costs only a couple of pennies, and it can detect Ebola virus. And each one of those circles is actually a distinct nucleic acid sensor for a part of the Ebola genome. On the right, you have, we applied it also to Zika virus, and we developed one of the first nucleic acid sensor tests that can distinguish between Zika virus and dengue at the time. We also worked with Feng Zhang's lab to develop a CRISPR-based detection system. And so now we can use CRISPR to very sensitively detect nucleic acids, and also this can be freeze-dried into paper or any kind of microfluidic system. We've also applied this to biomanufacturing, as well as one of the most arduous environments, which is high school educational kits there, and it seems to work just fine. So all this is freeze-dried. Our latest paper, we applied this to the detection of COVID, where we integrated this on the inside of a face mask, where it was able to detect virus-laden breath aerosols, and we can detect that using a system that's completely capillary driven. It doesn't require any power at all. And we can detect it with a sensitivity that's on par with WHO RT-PCR levels at that time. We also have electrochemical based sensing methods, which we developed in collaboration with the Ingber lab as well. So these are our technical capabilities. We have a poster. Please come find me. I find it's more productive at the poster session, and we can chat then. Thank you.
Thank you very much. Next up, we have Ben Krasnow from Verily.
few years ago, there was a story on the BBC about a woman who could smell Parkinson's disease. And she enrolled herself in a blind trial and was able to pick out who had Parkinson's by smelling their T-shirts. And she was 95% accurate in determining who had Parkinson's. And the one person she was wrong about later developed Parkinson's. So there's all these stories about detecting disease with smell, and what smell is are volatile organic compounds. And so I'm here to tell you about how these can be used to detect pathogens in the air, detect if someone is diseased just by smelling them. Wouldn't it be great if we had a machine that could just smell someone or sample their breath and determine what diseases they had? So that's what we did. I run a prototyping group at Verily, which is also known as Google Life Sciences. And we have built this small machine on the right that is a cost-reduced version of a mass spectrometer. And for all the analytical chemists in the audience, this is not just a poor man's version of a mass spec. It actually is fundamentally different. And when you're dealing with complicated mixtures of gases, the ion mobility spectrometer can actually be better than a mass spec in certain cases. And so our primary focus is looking at things like Parkinson's, but we also did a COVID study and found out we could detect COVID by isolating clusters of different VOCs. And so an interesting thing to think about is if you're trying to sample air and figure out if there's harmful microbes, pathogenic microbes, those have to come from someone, a person or an animal or something, who's also exhaling these other volatile organic compounds. And you might actually have a much easier time detecting those other VOCs than the microbes directly. Okay, so here's the output of our machine, just to give you an idea of what this looks like. Each blob on there is a different VOC. And we're comparing our machine against a commercial piece of equipment on the right called a breath spec. Our machine is on the left there. This is a super, super cost-reduced version, 100 times cheaper than anything else out there, and it performs almost as well. So the idea is you could have these low-cost machines distributed and collecting data to do all this sort of work. We also built a portable version. So someone at our company asks for a smoke detector that could detect disease, and that's what this is. So on the left, that's a really super miniature version of our machine that's even lower than the $4,000 price tag we had on the previous slide. And then finally, I just wanted to show that there's a database of sort of chemical bouquets that different types of microbes are known to produce. There's a whole database. And you can use this combination of VOCs to figure out what microbe you're dealing with by querying the database. So I don't have a poster here, but I'd be interested in talking to folks here about sensor tech. And thank you very much.
Thank you very much. Next up, we have Sissy Liu from Metalmark.
Good afternoon. My name is Cece Liu. I'm CEO and co-founder of Metalmark Innovations. We are a Harvard and spin out, and over 7 years ago, we started doing R&D specifically to address problems on the nexus of climate and health. Next slide, please. So climate change is here, and we're living with the consequences. Last year, wildfire smoke blanketed much of the US, and wildfire season has kicked off this year as well. And this has a lot of implications on how we address and provide solutions to treat the air in the indoor environment. Next slide, please. And so wildfire smoke and poor air quality from the outdoors can infiltrate easily into the indoor environment, in addition to the accumulation of airborne viruses, pathogens, and fine and ultrafine particulates and volatile organic compounds. And these lead to a number of illnesses that are acute or chronic. And they affect our concentration and productivity. Next slide, please. And the current solution to all of these problems is by ventilating using HVAC systems. And this is really expensive. And HVAC ventilation contributes up to 20% of building energy use. And they are very capital intensive. At the end of the day, they provide a suboptimal solution to air quality. Next slide, please. And this is why we designed the Tatama, the smart, distributed, hassle-free air cleaning system. Next slide, please. And we start by providing effective air cleaning in the commercial building space. This is very much targeted towards schools, hospitals, and commercial offices. Recently, we were awarded by the EPA after testing our baseline technology and validating it. In addition, we provide value propositions by having very low maintenance. This is really important for customers. So let's go to the next slide, please. On top of that, we layer indoor air quality sensors and data And using that, we optimize performance of our system to balance between occupant health and also energy requirements. Next slide, please. Our system is very easy to install into a variety of architectural ceilings, including what we can do here, for example. Next slide, please. And we have customers in different spaces who really love our solution, including schools that have installed our systems. Next slide, please. So we have a poster, and I think our specialty that we bring to the table is really in the TA3 area, and also to integrate directly with BMS systems for HVAC controls as well. And so if you're interested, please come find me during the poster session. Thank you.
Thank you very much. Next up, we have Aditi Nig from Draper Laboratory.
Hello, everyone. My name is Aditi Naik. I'm a senior scientist at Draper Laboratory. Draper is a nonprofit organization with a core mission to serve the national interest through applied research and development. In particular, that means that we take very early stage concepts and that we can translate those to later stage, more integrated solutions that are also fieldable. We have our main location in Cambridge. We have a variety of smaller satellite campuses across the country. We have over 12,000 square feet of BSL-2 level space, as well as access to a BSL-3 level space. And in particular at Draper, we are very excited to have innovative designs, rapid prototyping options, system integration for a lot of new solutions, especially around microfluidics, nucleic acid detection, biomaterials, bioengineering. And in particular, we've also had a lot of experience working with different government contracts. In particular, we've had successful programs with DARPA, ARPA, BARDA. We've been in prime positions. We've been in subcontract positions. For the Breathe opportunity, we're looking to leverage our experiences at Draper to develop a high-performance air sensing, biosensing system. So we are looking for collaborative teaming for the TA2 and TA3 focus areas in particular. So specifically, if you're interested in looking at the respiratory risk assessment or healthy building models, building science, filtration, ventilation, these kinds of things, please reach out. We do have a poster as well. There are a key couple of focus areas that I want to point your attention to here that we've developed at Draper, including an automated nucleic acid extraction system. So this is a disposable cartridge. It accepts raw sample input for bacterial and viral samples and can collect and purify that nucleic acid. We've been able to design that fully internally in-house, and we've done it with amenable, scalable manufacturing techniques as well. Then alongside that, we don't just build the microfluidics, we also build the integrated systems that go alongside it. So including all of the components, the hardware components, electronic components, and other things like that. We have experience with ruggedization, miniaturization. Then recently on the IARPA Felix program, we developed technology that can do enrichment of nucleic acids using very low sample volumes, as well as using non-destructive testing methods in order to do that. And the goal of that project was to be able to look for markers of engineering, but can also be modified for whatever the final application is of interest. And then lastly, we do also have developed a fieldable air sampling system that is currently being deployed on the International Space Station. It uses differential mobility spectroscopy. And we've also been able to modify that to be able to test for fungal lung infections for patients as well. So as I mentioned, we do have a poster, so please feel free to stop by, either talk to me or my colleague Dan Cunningham-Bryant, and we look forward to working with you. So thank you.
Thank you very much. Next up, we have Chris Scabu from Gate Scientific.
Thanks to everybody and to the Breed team. This is a great opportunity for us. I'm here to tell you a little bit about our air sampling platform. Next slide, please. We are located here in the Bay Area, in Milpitas, about 30 minutes south. We are product developers. We make stuff. We make money by making stuff for us that we commercialize. We make stuff for other people, and we make stuff, technology, and then license it out. We are very experienced in reagent, consumable, and instrumentation developer. We're co-located with a contract manufacturer, and we can handle development from concept stage all the way through low volume manufacturing. We can do regulated environments as well. Next slide, please. So this is a system that we started developing about halfway through the pandemic. We're calling it the BioAries. We see this as a TA1 solution. This is a fully integrated air sampling qPCR platform. It's completely autonomous. It has wireless communication. The air comes in the top at 100 liters per minute, gets shot through the filter there. It collects any pathogens. The instrument then seals around that, and we perform qPCR right on this consumable that has dried, stabilized reagents as well as the wet reagents in there. We've shown that we can get detection down to about 33 genomic copies per cubic meter. We really appreciated somebody from Virginia Tech talking about how noisy these things are. So we spent a lot of effort making these things not noisy. So we're able to get the thing running down about 50 decibels so we could run it in this room, still have a conversation. So the consumable at volume, we can get down to about $5 a test. Currently, it's 4-target multiplexing. And maybe I'll get into the next slide and we can talk a little bit about that. So this is an actual example. Once you form the chamber around the filter and do the PCR, we can do 4 colors. Here's a 3-plex. And this is, the actual camera images as it goes through the PCR cycle, so you can see it lighting up. Next slide, please. So this is really cool. This was some data we did with a prototype towards the end of the pandemic where we compared nasal swabs. We put the instrument or a breadboard of the instrument into a patient's home who had COVID. Took nasal swabs, 0, 1, 2, 3 days, looked at the COVID signature as well as the RNase-P in the pink. So the RNase-P was constant on the left there. And then on the right, we actually had our system running overnight. And you can see that the RNase-P stays constant, and we actually pick up the decay of the COVID signal and similar ratios as the nasal swabs. So this was really exciting data. Next slide. How are we going to multiplex? So we've developed a TACMAN array system, and this allows us, and here
We are over time for this talk. Sorry about that.
I'm sorry.
But thank you very much, though.
Yeah, thank you.
Yep. Next up, we have Braden Stump from Handex.
Hi, my name is Braden Stump with Handex Scientific, and today I'm here to talk to you about our water condensation growth tube collection technology. Next slide. So getting right to it, we are looking at technical area 1 biosensors, but what we foresee our role being in this is a gateway between the bioaerosol of interest that we are all looking to capture and capturing them very efficiently across all sizes and types, and bringing that as a concentrated intact sample to a wide variety of biosensing technologies, so improving your limit of detection. Our unique technique has 3 primary advantages that I'll be discussing. The first one being a very efficient capture of particles across a wide range down to 5 nanometers, which is extremely low, right? Up to 10 microns. Additionally, we have gentle capture of bioaerosols for virus, bacteria, and other microbes, gentle enough to either capture viably or keep genomic material like DNA, RNA intact at a very high rate. Finally, the third advantage is samples are concentrated and convenient for passing down to things like microfluidics or other techniques ready for analysis. Next slide. So the technology itself, we licensed from a company also right down the street, Aerosol Dynamics in Berkeley, who some of them are here as well. And it effectively, as aerosols coming through, utilizes supersaturation to create this fog of micro droplets around aerosol in the flow stream to condense water around particles as small as 5 nanometers. This reduces thermal shock. This allows you to capture more efficiently, and it allows you to capture particles more gently than other techniques. Next slide. So going through the advantages, this efficient capture over all particle sizes really starts to shine at lower sizes, which is where you see a lot of viruses in the air. So what you're seeing is our water condensation growth tube collection technique compared to 2 other industry competitors. And as you can see, not only do we have 90% efficiency and up regardless of liquid or solid substrate, it's also consistent across this wide range as well. So virus, bacteria, fungi, et cetera, we are confident in our ability to capture it. Next slide. You can also look on our website and see over 40 publications on our technology, many of which are about viable or gentle or genomically preserved capture of our technology. Plenty of folks here have published on it. Lindsay Marr's lab, this Hawks et al. Paper had really cool with our smaller sampler data with infectious SARS-CoV-2 capture. C.Y. Wu's group with Lednicki et al. John Lednicki's group as well. Published a really cool study utilizing viruses in a hospital room and being able to capture live SARS-CoV-2 and infectious SARS-CoV-2 from that, as well as Mark Hernandez's very cool school study paper as well, which is on my poster, but not this slide. Next slide, please. Final thing, I'll blow through this. Sample collection is really customizable, and we are a team of engineers who want to make it happen for automated techniques. Liquid vials with ports in the bottom going to microfluidics are possible. Solid wells that are time resolved, microscopy substrates, swabs for things like sequencing. Thank you very much. We're ready to customize. Come see me in my poster.
Thank you very much. Next up, we have Suresh Panthi from Gen Endeavor.
Hi, I'm Suresh from Gen Endeavor, and why we all are here is we consider that breathing clean air is a fundamental human right, and unfortunately, the air is contaminated with aerosols with microorganisms that can cause infection, and we need to find a real-time solution for this one. So, next slide, please. In this line, where we are is we are focusing on the detection of nucleic acids in the air. And our input will be on TA1. And what we have developed is two strategies or two things or two technologies that do not use enzyme and that can amplify nucleic acid. First one is autoligation chain reaction. This is by using a chemical reaction, we can amplify the signal, and we need the thermal cycling procedure for this autoligation chain reaction. And another one, we use isothermal chain reaction. So by using chemical reactions at isothermal or constant temperature, we can detect the presence of nucleic acid. Next slide, please. By using these two strategies, so our focus has been on air monitoring, tuberculosis, ovarian cancer detection, and sepsis detection. And what we can bring or what we are working on air monitoring side is our current approach includes the air sampler that is like more than 5 times more efficient in terms of air sampling. And then we have multiplex detection using the portable system. And we are using a biosensor that uses the laser light for the detection. So by using these strategies, what we think is like we are trying to monitor air through these strategies combined with our ICR approach. So based on this, what I say today is we will be good for TA1, and then we are looking for partners to working on TA2 and TA3. Thank you.
Thank you very much. Next up, we have Patrick Girding from R0.
Good morning, and believe it or not, you're almost done. I think there's one person after me. I'm Patrick Girding. I lead the public sector at R0. We are excited to talk to you about TA3 a little bit, but we also have slices of competencies in TA1 and 2. Next slide, please. If you like data like I do, you know who this guy is. If Mr. Deming was here today, he would be hearing us talking about indoor air quality, and he'd remind us that it doesn't just happen. It has to be built in. And we've been talking about that a lot. R0 has been doing that a lot. We've been building in air quality in indoor environments through monitoring, forecasting, and actually improving the health and quality of indoor environments. Across our installed client base of education, healthcare, commercial buildings, government buildings, detention centers, all sorts of senior living facilities, we are bringing data in from a variety of sensors and from our sensor portfolio to do data visibility and provide that data to real world software that is doing rules-based analysis of that data to create a management platform that's open and integrated and can drive change and action in ventilation, in disinfection, in space planning, and more. Our product portfolio and our technology portfolio starts off with a variety of sensors that gather all sorts of vital information about the indoor environment in order to act on, whether that's occupancy in space, whether it's temperature, humidity, indoor air quality, things like particulate matter, volatile organic compounds, and more. We use all that data to inform and educate us. We also offer a host of UV disinfection solutions that have sensors built in as well. And that ultraviolet system can actually inactivate the pathogens that are at risk that are causing risk in the indoor environments, up to 99% and higher of SARS-CoV-2, flu, RSV, et cetera, in the occupied and unoccupied spaces that we all exist in. Lastly, all that data is gathered in from all of our sensors, and it's brought in and analyzed by customizable real-world solution and software that can actually drive actionable insights to BMS systems, to people, to disinfection systems, to make educated and clear choices about how to move forward in managing our building and making clean and healthy indoor air. The most important part is we do it all with no retrofits. 80% of the buildings that exist today will be here in 2050. We can't count on new buildings to be built. We can't count on putting millions of dollars into old buildings with monolithic systems. We've got to be able to do it now. I look forward to talking with you all in the poster sessions. Thank you.
Thank you very much. Last up, we have Anita Goel from NanoBio Sim.
Hi, everybody. Pleasure to be here today. So I'm Anita Goel, NanoBio Sim, and I think I'm going to pull this. So our focus is on highly sensitive quantitative nanobiosensors for detection that we've also integrated into mobile devices. We work at the nexus of physics, nano, bio, and also AI. Okay, so our company focuses on really creating ways to very accurately detect and quantify viral loads and other kinds of molecular signatures in a decentralized real-time way. We have very high sensitive nanobiosensors that we've integrated using microfluidic enabled platforms into mobile devices. That won the first X Prize. It's been previously FDA approved. We build multiple apps for multiple types of targets or diseases. And we've worked a lot with DARPA, NASA, FDA. And one of the things that for this program, I think we bring capabilities for TA1, ultra high quality, high resolution data acquisition. We're interested in partnering with people with very high quality air sampling systems to collect the aerosols that could be integrated with these kinds of nanosensors. We have also integrated these mobile devices in an IoT enabled way to do advanced data analytics. How do I do next slide, please? Next slide. Oh, there, never mind. So we have a FDA approved, very ultra sensitive at-home saliva collection system for COVID, one of the most sensitive systems. We're actually partnering with the FDA to do quantitative viral load on COVID. And next slide, I'm having some trouble with the slide. And so in our mobile devices, we've done Ebola, Zika, COVID, several things for the pandemic, resilience features. And let me try to go faster. I'm having trouble with the slide. We're part of the FDA digital diagnostic program. Next slide, please. Where we're actually using some of these tools with an ecosystem of academic healthcare centers and deploying them to collect various kinds of feasibility data on acute and long COVID patients using the quantitative viral load and connecting that with various advanced data analytics. So in summary, we bring capabilities in TA1 and TA2, but interested in partnering with TA3 and TA2 and people who could help in TA1 on the air collection and sampling. Thanks very much.
Thanks, everyone.